let's talk about different types of neurons. Neurons can be classified according to their structure or according to their function. So let's classify the neurons according to their structure. So types of neuron according to their structure, they can be unipolar neuron, bipolar neuron, or multipolar neuron. So multipolar neurons constitutes 99% of the neurons. And these are the different types of neurons: multipolar, bipolar, and unipolar neurons. So if you search on the internet and if you type neuron, you will see these multipolar neurons. Of course, it makes about 99% of all the neurons, and therefore most of the internet search will show you this type of neuron. But don't forget, we also have bipolar neuron and unipolar neuron, but they constitute less than 1% each, and therefore multipolar neuron is the most commonly seen or most commonly drawn neurons for us. So if we look at the multipolar neurons, we have our cell body here, and there will be dendrites, which are branching out from the cell body. The dendrites, tips, ends, they will act as a receptor and take all the information from the periphery to the cell body. And finally, that will generate action potential, and that action potential as an electrical impulse or Nernst impulse we carry along the axon, and it will go to the terminal endings, which are called terminal arbitraire or terminal boutons. This is multipolar neuron. The second type, which is bipolar neuron. If you look in the cell body, there are only two processes coming out from the cell body. Yes, all the dendrites here, multiple branches dendrites, they unite to form one single large dendrite, which carries the information from the dendrites, stimulus information, to the cell body. The cell body will process and generate electrical impulse, which will bring the nerve impulse or electrical impulse along the axon and come to the terminal boutons. And number three type, which is unipolar or pseudo-unipolar neuron. If you look at that, we have cell body, and only one process come out from the cell body, and the dendrites, which will unite and form here, comes to the cell body, and then from the cell body, another branch go out to the terminal endings. Well, the neurotransmitters will be discharged. And therefore, I hope you will appreciate the bipolar and unipolar neuron on top of the multipolar neurons. So let's look at the different regions of the neurons with relation to their function. So the cell body and the dendrites, they will receive the stimulus, they will detect the changes in their environment, and that information will be taken to the cell body. And as you remember, around the axon hillock, that we decide where the electrical action potential or nerve impulse will be discharged. And if it is discharged, it will be carried along the axon. And therefore, axon is a conducting region of a neuron. And finally, it comes to the end where the terminal boutons are. The terminal boutons will release neurotransmitters such as acetylcholine. The same thing for the bipolar neuron and unipolar neuron. There could be structural variation even on the multipolar or bipolar or unipolar. But what we have to be aware is multiple dendrites come to the cell body. From the cell body, one axon come out. The same thing, multiple dendrites come to the cell body. From the cell body, axon come out. That is the multipolar neuron. And in the bipolar neuron, of course, there will be many dendrites which will form into one and large single dendrite here, and then we come to the cell body. Unipolar neuron, multiple dendrites, they fuse to form the peripheral process of the axon, and then they come to the cell body, and from the cell body, another central process, which is again axon, will come out. So multipolar neurons, they are usually found in the motor neuron. If you look at this diagram, this is the cross-section of the spinal cord. You can see the dark color, which we call gray matter, which is H-shape here, and the white matter, which is surrounding the H. And you can see the motor neuron, which is the multipolar neuron with the cell body here. It came out from the anterior root and ventral root here, anterior horn cell to through the ventral root, came along and came in, supply the muscle. So this is the multipolar neuron, which is found in the motor neuron. Usually, bipolar neurons are found in the eye, so they are dendrites which are present in the retina of the eye, come to the cell body, and the axon will go to finally go to the occipital lobe. The bipolar neurons are also present in the nasal epithelia, olfactory mucosa. So if you look at here, from the skin, the sensory impulse will be carried along the axon, and it comes to the cell body here, and from the cell body, it leaves again to the posterior root of the gray matter in the spinal cord. This is the anterior root, this is the posterior root, this is the anterior horn, this is the posterior horn of the spinal cord. And therefore, this is a type of the unipolar neuron, because it has the axon, which is coming from the skin, and it has the cell body here, and there is another central axon going from the cell body to the dorsal root. And this is called dorsal root ganglia. So you will see this when we talk about the peripheral nervous system. So let's classify the neurons according to their function. If they are carrying the sensory impulses from the periphery to the central nervous system, they will be called afferent neuron or sensory neuron. The opposite, if they are carrying motor impulses from the central nervous system to the periphery, the target organ would be like skeletal muscle, then they will be called efferent neuron. And there is one neuron which can exist between the afferent and efferent neuron, which is what we call interneurons. So we have talked about the neurons, different types of the neurons. We have classified the neurons according to their structure, according to their function. Now let's look at the neuroglia cells. These are the supporting cells which we protect and nourish the neurons. There are many different types of neuroglia cells. They are astrocytes, microglia cells, ependymal cells, oligodendrocytes, Schwann cell, and satellite cells. So as you can see here, astrocytes are the most abundant neuroglia cells. They regulate the neurotransmitter level, they control the ionic environment, they increase or decrease the blood flows in the capillaries in the active brain region, and therefore astrocytes play an important role. The microglia cells, which are micro, means small, they are least abundant and they are the smallest, and their job is for phagocytic function. And of course, because of the phagocytic function, they derive from the blood cells. Ependymal cells, you will see ependymal cells in the ventricles, which are the cavities inside the brain, which is filled with cerebrospinal fluid. Ependymal cells will secrete CSF. It is just an epithelial cell. We need to remember oligodendrocyte and Schwann cells. Oligodendrocytes will be present in the central nervous system, and Schwann cell will be present in the peripheral nervous system. Their job is to produce myelin, and that myelin will wrap around the axon. So they have the same function. If they are present in the central nervous system, they are called oligodendrocyte. If they are present in the peripheral nervous system, they are called Schwann cell. Satellite cells, they will surround the neurons, cell body in the ganglia. So that's posterior root ganglion. We talk about that. So these are the examples. This is the astrocyte here, which is the star shape. It's a large, it is most abundant. If you can see, it is going to enclose the capillaries. By doing so, they will control the vasodilatation or narrowing of the capillaries in the brain. They are going to the dendrites of the neuron here. So they have a big influence on the neuron. And this is the microglia cell, which are the small, least abundant, and their job is for phagocytic function. And this is the ependymal cell, which is just like an epithelial cell, might be looking like a columnar epithelium. Their job is to secrete cerebrospinal fluid. And we have to remember oligodendrocytes and satellites, Schwann cells together. Their job is to produce axon and protect. They produce myelin and to protect axons. So that's the astrocytes, which is the most abundant. Their job is to regulate the neurotransmitter level, to control the ionic environment, increase or decrease the blood flow in the capillaries of the brain, and they produce brain-derived trophic factors. So this is microglia cell. Again, I believe you know this already. Least abundant, smallest, phagocytic function. They derive from the blood cell monocyte. An ependymal cell here, they look like a columnar epithelium, and their job is to line the cavity in the brain and spinal cord for CSF protection. They do have little cilia here, as you can see. Their job is to circulate, to facilitate the CSF circulation. Oligodendrocyte, they are present in the central nervous system. They produce myelin. Oligodendrocytes here, you can see oligodendrocytes. They will produce myelin, and that myelin will cycle the axons of the nerve. And the same thing, like Schwann cell, we produce myelin, and Schwann cell will produce myelin, and that myelin will encycle the axons. So myelin sheath, why do we need myelin sheath? They are composed of myelin, which is lipoprotein. They surround the axon. We have seen that already. They act as an insulating layer for the axon. They prevent the leakage of the electrical impulse, which is running in the axon, and they increase the speed of the electrical impulse propagation and to make it more efficient. So there are two ways, like myelinated fiber axons and non-myelinated axon in the peripheral nervous system, like myelinated axon, if you look at that, that will be surrounded by the Schwann cell plasma membrane here. And then if you keep looking at that, one single axon is multiply surrounded by the myelin sheath here. And non-myelinated axon, yes, they still do have some Schwann cell, but they are loosely covered or protected by the Schwann cell. So it is very different from the myelinated and non-myelinated. Myelin sheath in the peripheral nervous system, massive but concentric layer, they are tightly packed, and there are gaps in between the myelin sheath layer. These gaps are called node of Ranvier. And nerve impulse, they do not travel along the myelin covered axon. Instead, they will jump, and that will increase the speed. What am I saying? That here, the nerve impulse is coming along the axon. If there is a myelin sheath here, Schwann cell forming the myelin sheath, the nerve impulse, instead of going like that, it will jump from the one node of Ranvier, which is the non-myelin covered axon site, here to there, here to there. Because of this saltatory propagation of the electric impulse, it increases the speed of the conduction and it is more efficient. So I want you to have a look on this website. It is from the brain tumor, like information website from the Johns Hopkins University Pathology Department. And the reason is now, I believe you know what is our brain is made of. The brain is made of neurons and neuroglial cells. The brain will be covered by the meninges. And therefore, if someone has a brain tumor, or in other words, to be more precise, intracranial tumor, it will be coming from the meninges, it will be coming from the neuroglial cells, but it will not be coming from the neurons.